So let us discuss the chapter cell cycle and cell division. Okay, so cell cycle and cell division. So before getting into the topic, let us understand what a cell can do. A cell can divide, a cell can elongate, and a cell can differentiate. A cell can divide, a cell can elongate, and cell can differentiate. After the differentiation, what a cell will do? A cell will mature, right? A cell will mature. After maturation, what happens to the cell? It will perform a specific function, right? Perform specific function. It will perform a specific function. After performing a specific function, what they will do? They will the lifespan occurs in the cell. Lifespan occurs. Lifespan occurs. After lifespan occurs, what happens? The cell death. Right. Lifespan occurs means the cell death. So then term used for the cell death is known to be apoptosis. What it is? Apoptosis. So apoptosis occurs for those cells which attain the lifespan after the differentiation. Okay, so here what we are going to discuss is how the cell divide. Okay, so before that we need to understand what is a cell cycle. What is a cell cycle? A cell cycle means it is a sequence of events. It is a sequence of events. Sequence of events. Which and all events? That is, first it will be, that means the cell will duplicate. Cell will duplicate. Cell will duplicate. In which and all cases they will duplicate? In the case of genome, the genome get duplicated. Genome get duplicated. Organelle get duplicated. Organelle get duplicated. Okay, and they get divided into two daughter cells. Two daughter cells. Okay, so it is a sequence of events. Sequence of events. What is cell cycle? It is a sequence of events where the cell will duplicate its genome, its organelle, and forms two daughter cells. So that is the cell cycle. So cell cycle, if you say about the cell cycle, it has to go through two phases. One is interphase. One is interphase. Second one is M phase. Interphase and M phase. Interphase and M phase. So if you say about the interphase has to go through different phases. That is, the interphase move through different phases. The first phase will be G1 phase, second phase will be S phase, and third phase will be G2 phase. Okay, so let us write the phases. G1 phase, S phase, and G2 phase. And M phase also has to move through different phases. So first one will be the karyokinesis. First one, karyokinesis. So in the cell division, the first process take place is the division of the nucleus, then only the cytoplasm. So cytokinesis after the karyokinesis. Okay, so karyokinesis means nucleus division. Nucleus division. Nucleus division is known to be the karyokinesis, whereas cytokinesis means cytoplasm division. Cytoplasm division is known as cytokinesis. Okay, and this karyokinesis need to be go through different phases. That is, the first phase for the karyokinesis is prophase, then metaphase, then anaphase, then telophase. Last one will be telophase. Okay. First one is prophase, metaphase, anaphase, and telophase. Anaphase and telophase. Okay. So, in cytokinesis of plants and animals are different. In the case of animals, it is called centripetal, whereas in the case of plants, it is called centrifugal. Okay, centripetal and centrifugal. So centripetal means in the cell, the cytoplasm moves inwards to make two daughter cells. That is, here the nucleus are there, then it will forms to two daughter nuclei, whereas in the case of plants, the first that inward to from the inward to the outward, like this. Okay, inward to the outward. So this type of division in the plant cells is known to be centrifugal. Okay, so they will form a cell plate, and from the cell plate the two get divided, then the new cell wall will be formed, middle lamella will be formed, which is made of calcium pectinate. Okay, calcium pectate. Okay, then so that are the process phases of M phases and interphases. Now let us discuss about in detail about the interphase. Okay, so for that we require a circle. To show the cell cycle, we need a circle. So we have the cell circle. Now we need to draw the G1 phase. Then the S phase. Then the G2 phase. Okay, so these are the phases. The first one is G1, second one is S phase, third one G2, last one is M phase. So in the G1 phase, it is also known as gap phase 1. It is also known as pre-M phase or pre-mitotic phase. Post-mitotic phase. Because G1 comes after the M phase, right? So post-mitotic phase, or also we can say that pre-synthesis phase. It occurs before the S phase, so pre-synthesis phase. So these are the three different names for the G1 phase. Then what and all happens in the G1 phase? The first one is ATP production increases. ATP production increases. The second one will be cell growth. Cell size increases. Cell size increases means cell cytoplasm increases. Cell cytoplasm increases means the increased due for abunding the organelles, right? So the organelles, maximum number of organelles will be start to duplicate or produce. Then ribosomes, sorry, RNAs will be produced. RNA will be produced. Okay. Then protein will be produced. Protein will be produced. Then what? Nucleotides. Nucleotides. Nucleotides will be produced because the next phase will be synthesis phase, synthesis of DNA. So that for that it requires the nucleotides. Okay. So synthesis of nucleotides occurs, and in this phase cell is metabolically active. Cell metabolically active in this site. Metabolically active in this site. Okay. So the cell size increases, cytoplasm increases, and one of the most important point you need to remember is one is cell metabolically active. Second one is it is the longest phase in the cell cycle. Longest phase in the cell cycle. Okay. So these are the main points that you need to remember in the G1 phase. Then the next one will be synthetic phase. S phase is also known as synthesis phase. Synthesis phase. Synthesis phase. Okay, S phase is also known as synthesis phase. Why it is called a synthesis phase? Because it produces, it synthesizes DNA. It synthesizes DNA. If DNA is produced for the package, it should produce the histone protein also. So histone protein also produced. Histone protein also produced. Then what? Then the centrioles duplication. So in which phase the centrioles duplication occurs? Centriole duplication occurs in the S phase. So these two are very important points in the S phase. The first one, that is the synthesis, it is known as synthesis phase. It is synthesis of DNA. Then centrioles duplication occurs in the S phase. Centriole duplication occurs in the S phase. The next phase is G2 phase. In the G2 phase, what and all happens? In the G2 phase, the tubulin protein is produced. Tubulin protein is produced for what? For the spindle fiber formation. Spindle fiber formation. The tubulin protein is produced in the G2 phase. And in the G2 phase, what and all happens? ATP production will increase. ATP production will be increased, and centriole preparation. Here only the duplication of centrioles, right? Duplication. And here what happens? The centrioles preparation. Centrioles preparation. Also, one thing happens in the G2 phase is that after the S phase, the mitochondria, which has exon DNA, mitochondria plus Golgi complex, chloroplast, these will doubles in the G2 phase. And what are the name for this G2 is that first one is gap phase 2. Gap phase 2, that's the first name for this. Second one will be pre-M phase, pre-mitotic phase, because it comes before the mitosis phase. Third one, post synthesis phase. Post synthesis phase, that is it comes after the S phase. So these are the name for the G2. Then name phase. If we discuss about the M phase, The M phase has two phases as I already told you. So first one is karyokinesis. Karyokinesis. What is karyokinesis? The division of nucleus. Then the cytokinesis. Cytokinesis. So karyokinesis also four phases. First one is prophase, then second one prophase, metaphase, anaphase and telophase. So this one is the M phase. M phase also known as mitotic phase. Mitotic phase where the daughter cells are produced. Okay. So this is all about the cell cycle. Okay, G1 phase, S phase, G2 phase and M phase. And one of the most important point that you have to remember here is that after the M phase, the cell will goes to the G1 phase. The cell will goes to the G1 phase. But some of the cells will not further proliferate. So these cell will goes to another stage that is known to be G node phase. That is known as G node phase. So what is G node phase or quiescent phase? Quiescent phase. So quiescent phase or G node phase means it is a phase where the cell is metabolically active. Here the cell is metabolically active but they are inactive. Okay, but they are inactive. So in G0 phase the cells are metabolically active but cells are inactive for further proliferation. Okay, but whenever it is required, whenever the cells are required by the body, it will again move to the G1 phase. Okay, so whenever it is called back, whenever it is called back, it will start from the G1 phase. Okay, and start to proliferate. So this is the cell cycle which has G1 phase, S phase, G2 phase and the EM phase. Okay. Now let us discuss about C value and N value. So C value means it is the total DNA content, in a cell, okay, whereas the N value means total number of chromosome, in a cell. Okay, so what is actually mitosis? Means mitosis occurs in which cells? In the somatic cell, and it is a type of equational division. Okay, it will be same as the parent cell. The formed daughter nucleus cells will be same as the parent cell. So no variation occurs in the mitosis. That is no crossing over or no recombination is occurring in the mitosis, and it is an equational division. But after the mitosis, the 2C value become half. The 2C value, that is the DNA the value, the total DNA content in the cell will be reduced into half. How? That is, first in the G1 phase, the DNA, what happens to the cell? The cell size increases, everything will be increased in the G1 phase. But during the S phase, what happens? In the S phase, the DNA start to replicate. DNA start to replicate. So this is the DNA, and by the enzyme there will be a cut which opens the DNA, and from here it start to read the DNA and make a copy in each strand. Okay, in each strand an extra copy will be formed. So we know that DNA is a double stranded structure. So in each strand one new DNA will be formed. So an extra copy of DNA is formed in the S phase. That why the S is known to be the synthesis phase. Synthesis phase. Okay. Okay, if the chromosome number here were 1, if the chromosome number were 1 and the C value will be 1, but from here the chromosome number will be same, but the C value, the DNA, the value of DNA become 2, that is doubled. It is doubled. But in the G2 phase also same, the number of chromosome will be 1 and the C value will be 2. But after the M phase, okay, so up to this it was a one cell, but after the mitosis 2 cells are formed. So what happens? The DNA number will be halved. DNA number will be halved, but the chromosome number remains the same. Okay, so in the mitosis 2C value become half, whereas the number of N value or the number of chromosome remain same in the mitosis. Okay, so these are all about the mitosis. So once more, if you draw a chromosome, so this chromosome contain DNA, and this DNA after when it gets to the S phase, so this one is G1 phase. When it gets to the GS phase, what happens? A sister chromatid will be formed. Sister chromatid will be formed to occupy the another DNA. Okay. So the DNA number that is C value get doubled. C value get doubled, but the N value is same. N value will be same. Okay. So that is all about the cell cycle. Now let us move on to study about the mitosis.